Okay, we're gonna get it started. Um, you all turned in your assignment one, and hopefully you'll have grades. I think in the next few days, some of you may have gotten feedback. Can you see the feedback on it already? Or no, you can't see anything, right? Okay. Yeah, we'll publish them in the next few days. It looked like that many of you did well, even in the first attempt, but we're gonna give you the chance to resubmit. You'll get feedback from the TAs and then you'll resubmit to make it better. But I think the first grade already looks pretty good across the board. Um, any questions about that? I think the assignment was a straightforward, and we did provide you with templates and stuff like that. So the expectation is that you have followed those templates and read the papers. And any difficulty with reading the papers? Did you feel more comfortable after a few weeks, maybe reading the papers? And do you see any improvement in your own ability to process these sorts of texts? I think something I mentioned, really one goal of this course, or maybe the main goal of this course, is for you to become comfortable reading papers, right? A lot of you have aspirations to continue in academia or become clinicians, and that requires you to, read articles, write articles. So it's important to know the basics and be able to be comfortable just picking up any paper that is relevant to you and not be hindered by not knowing the basics of the brain. Um, so that's something that we're pursuing and hopefully we can achieve. Um, any questions? Yeah. They are already on the syllabus. There are a bunch of papers. You could pick one of them and do the assignment too based on that. Yeah. Does it sound good? I'll do that. Does it sound good back there? Yeah. Okay. So there is some noise, but um, so we left off here. We were talking about the brain, and I wanted you all to go and look at some models of the brain to become comfortable of when you look at the brain, you know where you're looking at. Hey, no, I muted it. Huh. Can you hear me over the birds? No? Why does this happen?
This happened last year in this room, and we did not figure out a way to fix it. I came, looked. Oh.
Yeah, well, we'll restart it and then hopefully. But what does it restart exactly? Does it restart the speakers too?
Try to get touch with someone from behind. Or everyone can move closer. We can turn the microphone off. I think it, yes, the microphone. If you turn the microphone like down or off, I think the first
What's that?
if you turn the microphone off or down,
Yeah,
the bird, yeah, the bird.
if I mute the microphone, then it's just my voice.
Right. I know. It drove us up last year. Yeah, but this isn't always like that.
So let's get in touch with the IT. But yeah, we should all just move down. Move to I mean, you could fit everyone in the first probably 4 rows. Okay, we can move everyone to the first 4 rows. Is that not clear? What's that? Just come down. So it feels a lot cozier and nicer, so that's okay. Everyone can hear me now? Yeah? All right. We'll do it without the microphone then. So we left off here. We were talking about the um the brain, and as I said, I wanted you all to look at some models of the brain. I'm just gonna pull up one that I found. Am I sharing my screen? Yeah. Which I thought was very nice in terms of showing the brain and just trying to give you, if I go up there, the brain. Okay, so what I wanted you to be able to do is that look at the brain, like, and be able, when you see a picture of the brain in a paper or in the book or in the slides, to know what you're looking at, right? So we're looking at the brain here. This is the cerebellum, and this is the brainstem, and that tells you that which way is this person facing? Left, right? Because the brainstem and the cerebellum are in the back. And we're looking at which hemisphere? The left hemisphere, because the person is facing that way, so you're looking at the left hemisphere. Now, if we turn this around, I managed to do that, yeah. So if we turn this around, this is the left hemisphere. You can imagine if I'm looking at it from the other side without the right hemisphere being there, it looks something like this. Here you still see the brainstem and the cerebellum in the back, but you could see that the left hemisphere would look different because you're looking at it from a different angle without the right hemisphere being visible there. If you're looking from the back of the brain, let me go back again to the whole brain. There's a what?
They're in a sense of number.
Okay. If I'm looking at the back of the brain, it would look something like this, right? The left hemisphere, the right hemisphere. If you're looking from the top, it would look like this. Another sign, the front narrows down quicker than the back. So if you just, let's say, look at the right hemisphere, right? You can imagine if I'm looking at it from down here and from this angle, I would see something like this. What would be this area? It would be the temporal lobe. This would be the occipital back here, and it would be the frontal. It's all, I'm looking just at the right hemisphere. This is something that I was showing here in my slides. This is the figure that I was showing.
Microphone's feeding back. Weird.
So let me stop. Okay, so this was what I was showing last week. Here, I'm showing the right hemisphere, but we're looking at it from a very specific angle. This is the angle we're looking at. So we're looking at the right hemisphere, but we're looking at pretty much from this angle. So you could see the temporal lobe down here, the frontal lobe here, and back here is the occipital lobe. And the visual cortex is back here. and that's this figure. The other difference is that metaphorically, we're putting air in the brain in order to open up all the creases and the folds in order to turn it into the cortex into a surface. So that's also the other difference from, let's say, this structural that you see here. But go look at this model and become more familiar, as I said, with different views of the visual cortex. As I mentioned last time, cortex is a surface, basically a two-dimensional surface, the size of a large dinner napkin. It's bunched up, and because it's bunched up, you see this is it appears this way as if it's folded, and there are different depending on their gyri and sulci, as they call them, but don't worry about that. So because it's a surface, it really is not just one thing, but rather it's like it itself is layered. These few millimeters of the cortex or themselves have multiple layers. And people have identified 6 layers, with layer 4 itself having 3 sublayers. So layer 4 is divided to A, B, and C. And as I said, when we talked about this visual pathway, the geniculostriate pathway, the signal from the eye goes to lateral geniculate nucleus, LGN, in the thalamus, and then it projects to layer 4C. So all the input from the eye goes to LGN. From the LGN, all of them project into this specific layer of the cortex. This layer of the cortex, as I said, this is just in the thickness of the few millimeters, it itself is layered. So when we're looking here, this is the thickness of the cortex, and this a little thickness has itself 6 layers in it. So imagine it's a very thin dinner napkin, but 6 of them stacked on top of each other. Okay. So the type of cells that we get in this specific area, the layer 4C of the primary visual cortex, they also have the center surround antagonistic receptive field that we talked about, and you're kind of familiar with for now. Then there are cells with more complex receptive field, or in this area too, but in different layers of the cortex. And we saw, I think, simple cells, but we're going to talk a little bit more about complex and end-stop cells. So the simple cells that we saw are these. You had them on the exam, I think, right? We talked about cells where there is an elongated excitatory area in the receptive field, and it's surrounded by inhibitory areas. So it means that so this whole thing would be the receptive field of the cell, because the cell responds to the light in this area. But if there is light only in the excitatory area and not in the inhibitory area, that's when the cell fires the most. It is an antagonistic receptive field also. And if you remember, we talked about there are 2 types of these cells. There could be on center, off surround, which means that the center area, if it is excitatory, the surround area is inhibitory, or off center, on surround, which is the center area is inhibitory and the surround area is excitatory. So you could imagine that different patterns of light would activate these cells very differently, depending on what they are. But we talked about this, so I'm going to go through them quicker. If this is the pattern of light, and you have a receptive field like this, well, you're gonna respond much more strongly when the light is vertical and only in the excitatory area of the receptive field. If the light is horizontal, the cell would not respond so much because the ratio of inhibitory to excitatory is higher. And then when the light is in this shape, you get something in between. So the response of the cell in this case really depends on how much excitatory input it gets versus how much inhibitory input it gets. And then that could define the response of the cell. So you're all familiar with these because you've seen them before. So I'm using another term here. I'm saying respond to stimuli having a particular spatial orientation. They are orientation tuned. What does that mean? When I talk about a cell and I'm saying they're orientation tuned.
So I would expect to go up if like there, the cell would be like vertical, I guess, right?
So let's clean up the language. A cell is not perceiving anything, right? The cell has something in its receptive field and it responds to it, right? So perception is not a property of a single cell. Just the language that is important that you know about, like, right? There are terms that I use and I'm going to use them interchangeably. I said, okay, this cell is tuned for that, or this cell likes that, or this cell detects that input, or all of these things mean the same thing. What does that mean? Somebody else? Yeah. It means that this cell fires very strongly when that specific input is in its receptive field. That's what that means. When it's tuned for something or when it likes something, it means that it's going to fire a lot. It's going to be maximally excited when it sees that thing in its receptive field. Well, if it's not in the receptive field, the cell doesn't care about it because it doesn't have access to it, right? When it's in the receptive field, it means that there are connections, multiple, it could go through a cascade, but there are connections that connect those photoreceptors to this specific cell. So that's its receptive field. And if there is this input in its receptive field, it's maximally active or is tuned for it. So this would be a bad stimulus for this cell for the reason that I was talking about, is that this is, again, it's supposed to be light, but it's in dark. So it's supposed to be how the pattern of light looks like. You could see that it activates inhibitory areas of the receptive field on either side of some excitatory. But the relationship is that, well, there's more inhibition of the cell than excitation, so the cell wouldn't respond to it. Having this receptive field, having a connectivity that results in this receptive field, right? Receptive field is just an abstraction. You're just showing what this cell likes using this receptive field and using this schematic. But what you're communicating is that, well, there is a connectivity, underlying connectivity, a circuit that controls the response of this cell, and that control of the response follows this receptive field. That if there's light in this part of the world, the cell responds to it strongly. If it's outside of that, it gets inhibited within its receptive field. So we also talked about this, that there is a cascade, right? That there has to be some cells that have simpler response that are connected to this cell for this cell to have its response, right? It doesn't come out of a vacuum, right? For a cell, for a simple cell to have this receptive field where light in this area inhibits it and light in the center excites it, it has to be connected to simpler cells, the center surround antagonistic cells that have excitatory center inhibitory surround in certain organization for it to inherit this response. Why? Well, if there are, imagine that these cells are tiling this space, right? So, and I'll talk about this in a little bit. The cells that are center surround antagonistic are in the eye. These are the ganglion cells. They're in the eye, so they're everywhere in the eye. If they're everywhere in the retina, it means that they're processing the light from everywhere in the visual field. So you have these responses everywhere in the visual field. It just so happens, or the connectivity for this cell is that it's connected to the ones that are aligned. And because of that, it inherits, as I say, this response because of its connectivity. Does that make sense to everyone? That there's a cascade of these processing, and the way you're connected defines the way you behave. Okay, we discussed this, so I'm going to go through them quickly. All right, so the cells can function as feature detectors for oriented edges and bars. Again, this is another conceptual point. What does it mean for them to be feature detectors? Mm-hmm.
To get detect details within stimulus.
Yeah, so what does detection mean in this case? What does it mean in the brain if I say these cells are feature detectors? Okay, so they can, so if I said, if there is a bar, a light bar in a dark background, there must be a cell in the brain that responds to this. Because these cells are tiling the space and have all sorts of different orientation selectivity, different receptive field sizes, different parts of this space. Does that make sense? So there must be a bunch of cells that really like this response. There must be a bunch of other cells that like this image. It means that there are a bunch of cells that maximally respond to this input. There are a bunch of cells that maximally respond to that input. So their role in the brain is that, well, I've detected something. I've detected a horizontal bar in this part of this space, and you let the rest of the brain do its thing, right? There's a higher level where another cell is looking at these oriented bars. But this cell's job is to say, okay, I've detected a horizontal bar in this part of the space. Maybe there's another cell that detects another bar in this part of the space. Another cell detects another bar in this part of the space. And then collectively, another cell is connected to them, and that cell could detect maybe a letter A. Does that make sense? So a bunch of cells are building the foundation of recognition by detecting something, by seeing, okay, I'm responding. Again, detection is, in this case, is a metaphor for what they do. Okay, these are simple cells. We talked about them. You did the exam, and you're kind of familiar with them. Any questions about simple cells? The simple cells are in the V1, and I'll talk about it. They, as I said, they tile this space, and that's an important point that I will explain more after we go through a couple of things. So also there are complex cells in V1. These are usually tuned for both orientation and direction of movement. So you could see a response here, for example. So you have an orientation here in the receptive field. And this orientation, the bar is when it's moving in the right direction, the cell is responding much more strongly to it. The cell is responding much more strongly to it. When it's moving in the left direction, the cell is not responding that strongly. So if you remember, these are action potential. This is what we call a raster plot. And each line shows an action potential. So the cell not only is only selective for a certain orientation, it's also selective for the direction of movement. So it's tuned for both orientation and direction of motion. Receptive fields are slightly larger, so they're not so specific to an area, but rather larger than that. So they respond to, because they have to respond to the direction of motion, they would have longer receptive fields. But if the light is not in the pattern of in the orientation that they care about, they usually don't respond at all. So orientation is very important. Direction of movement is additionally important, but not as important as orientation. These are the video that I showed. I didn't show the whole thing, but there was a video that I showed about people experimenting with different light patterns and their direction and their size. And it came out of that research where they found that, well, if you move the light in a certain orientation, in a certain direction, while certain cells respond stronger to it. Okay. So you could also imagine how a cell would, what kind of connectivity these cells would have in order to respond that way. Well, if a bunch of simple cells that are responding to the world that are next to each other, and you could imagine the cells that have receptive fields that are next to each other in the world are next to each other in the tissue. What is that property called? Retinotopic mapping, right? The cells that I was saying, if the receptive fields are next to each other in the world, these cells are next to each other in the visual cortex. That's called retinotopic mapping, that this mapping exists. So you can imagine that these cells are next to each other, and if they're all connected to a complex cell, you can imagine these complex cells would care that it's only a certain orientation it responds to, but also if it moves in this direction, it would be also responding to it. Again, it's not as simple as this. There are delays in these, and in order for you to capture direction of motion, we're not going to talk about it. But in order to capture direction of motion, you need delays in these connections, and there are delay circuits and those sorts of things, and people have mapped them out. But imagine the coarse connectivity would be that the complex cells is connected to simple cells that have receptive fields next to each other, and they all are in the same certain orientation. Okay. Questions so far? Yeah. Yeah, so if I had a pattern of light, like a, imagine it's an elongated bar, and then I'm moving in this direction or that direction. Just a simple direction of motion. So these are motion detection cells. So I'm gonna go deeper into the idea of feature detection and feature analysis. This is important in understanding the visual cortex, but also when we talk about object recognition next week, it's going to be very important to understand what this is. A lot of people, when they think about primary visual cortex, and it's debatable, when they talk about primary visual cortex, which is the area in the back of the brain, they think of it as a system for feature detection. So what does that mean? Well, it means that in order for you to act in the world, you have to know what's where. What is where? And that's what the visual cortex is going to give you. Well, there's a bar here, there's a person there, there's a car moving in this direction. Does that make sense? All of these things, you need information about them in order to act in the world. You need to know what is where in order to reach for it, in order to avoid, colliding, in order to run away from a predator without running into a tree, for example. So the visual system needs to give all that information for you to act upon. So we're going to understand the visual system in that sense, and in the sense of having a hierarchy that gives you all those important features, starting from very simple features. Okay. So V1, which is the first stage in the visual processing, something that we discussed last week briefly, was that there's something in V1 called cortical magnification. Cortical magnification, or foveal magnification, is that a relatively big area of V1, almost 10% of it, is devoted to analyzing info from a very small foveal region of the retina. So this is when you are looking at something, the input from that specific location is projecting onto the fovea. And we talked about that. So if I'm reading something, if we're all reading this text and I'm looking at this letter V, the light from V is getting projected into the fovea. And then that gets projected into the V1 through multiple stages. And when it gets projected to V1, it's magnified. I showed this briefly last week in this context when we talked about, let's look at this, for example. This is just a schematic, but let's look here. The distance between fovea, where it's shown by F, and 1 is really small. But you can imagine that a large part of V1 is representing this, the information in this area. So all this tissue that is here is representing this tiny circle here. On the other hand, you could see that the representation coming from further areas from the fovea that are bigger gets like kind of a smushed, collapsed. So you could see this here. Again, this is a schematic. If you're looking at this face of Newton, I think, when you look at, this is the point of fovea. When you look at the eye gets magnified. So all this tissue is processing the information coming from this part of the visual world. And then the hair and this part gets really small. So this is called foveal magnification. What I'm looking at that moment, it gets magnified. And all its features are being represented in all details. Like all edges are being represented, all the colors are being represented. When the areas that are farther from the fixation are not being equally as well represented because there's a convergence of information there, and then they get kind of averaged out. That's what I wanted to say. So this is the left part of the visual field. So this must be the right hemisphere, right? This is the right hemisphere, and we're looking at it. This is the right hemisphere. We're looking at it from that angle after removing the left hemisphere. Everyone gets what foveal magnification is? Okay, so that's what magnification is. This is just, again, a schematic of what that would look like. If this is the point of the fovea, you can imagine that a big part of the visual cortex would be representing what comes from the fovea. And then these angles are the angles of the visual world, as I was saying. Imagine this is positive 90, this is negative 90, because this must be 180, right? The world is 360. So the point that I'm looking at, the area around it is highly represented, and then the areas that are outside are being all kind of collapsed into smaller and smaller areas as you go out. So when you're looking at something, you have much more information about areas close to the fixation than the areas that get farther from you, from your fixation point, because the tissues are not, there aren't that much tissues representing those features. What that means, again, I hope you again get this more of a conceptual point of what representation is. Brain works with neural activity. If you want to act upon something in the world, it has to be represented in the brain. In what form? In what form would it have to be represented in the brain for you to act on it? What's that? What's the answer? Yeah. No. So I'm saying it's the word act, I think you're going with that, but I'm just saying, so for me to answer, is there a person sitting here to the left of me? If I want to answer that question, I have to have this part of the world in my head, right? In what form would that be in my head? Neural activity. This part of the world has to be represented in my head. That's what the representation is. Does that make sense? If I don't have everything is in your head, right? So when I'm looking, this part of the world has to be represented in my head, represented in my head in a form of neural activity for me to answer questions about it. Does that make sense to everyone? It has to be neural activity. Otherwise, if I'm looking this way, I'm not going to be able to answer questions about this part of the world. Why? Because, well, as I said, I'm representing up to this point probably, and then everything else on this part, I'm blind to it, right? It's not in my visual world. It's not represented in my head in order for me to answer questions about it. So when you're able to answer questions about something, it has to be represented in form of neural activity. So when we were talking about, for example, color perception, for me to be able to answer questions about, well, are these things different colors? What should be represented in my head? We talked about this, right? What should be represented? In order for me to be able to answer if these things are different colors, what should be represented in my head? Yeah, so the wavelength of the light coming from different part of this graph has to be represented for me to be able to answer questions about it. I have to sense it, and that sensation becomes neural activity, and then that neural activity is something that is represented in the brain, and then I can answer questions about it. If it's not sensed or if it's not represented, I can't, as a neural activity, I'm not going to be able to answer questions about it. Another important property of the primary visual areas is that they have a columnar organization. And this only makes sense if you understood what I meant by cortex being a surface. So the cortex is a surface when you blow it up, and you could now put an electrode in that surface and see what its properties are, and then move it a little bit, and then move it a little bit. We'll talk about what these columns are. So, what do I have after this? Okay, a bunch of these. So, when I said the surface of the cortex, if the cortex is just two-dimensional surface with a few millimeters thickness, what people have found is that neurons that are separated along this columnar organization, so neurons that are, if you put an electrode, this is the surface of the cortex. If you put an electrode and then record from a neuron, and then move it a little bit inside, record from another neuron, move it a little bit inside, record from another neuron, right? Because this is only a few millimeters, but there are hundreds of neurons from top to the bottom. And you could record from different neurons within this direction. The direction is, if this is the surface, the direction goes exactly through the surface. So in my graphics here, the direction would be what? The direction would be in this direction. Does that make sense to everyone? So again, you have to have a good 3D mental model right now. When you slice this and pull it out, you can imagine that all the cells are at the surface. So if you put an electrode and push it in this direction, that would go through the surface, or in this few millimeters. Do I have a better picture of this? Same here. So when you slice this and then you see that the cells are at the surface, you could actually put an electrode here and then move it through the surface, and that would be the direction that I'm talking about. So different location across the surface code for different locations in the space. So when you move it in this direction across the space, you're coding for different locations. However, all the cells at a given location are coding the same thing. This is an old figure that actually came out of that study that I mentioned. So what they found is that they're saying that when you go across the surface, when you go deeper and deeper in the surface, the response of the cells don't change. So it means that they're all responding to the same orientation of the bar. When you move from this location in the surface to another location in the surface, I'll go to this figure. This is a better figure. So when you go from one location in the surface to another location in the surface, then that becomes a different location in the space. Maybe I should have copied some of these figures so I wouldn't have to go back and forth. But the idea is this. If you are on this surface, if you go from one location to another location, you're representing, you're going to record the behavior of the cell responding to a different parts of the visual input. That's the idea. Okay, so what are these columns? So when you look at this, keep in mind, this is the direction of the depth of over the surface. So this is the few millimeters from up here to down here. This is the few millimeters that we were talking about. And the idea is that everything in this specific small column is representing the same feature. All right, let me step back. Look, what is a location column? A location column is a set is a tissue, part of the tissue that responds to the same location in the space. Now, if you go to the next location column, then you'll have a receptive field that is next to that. You go to the next column, then it's a receptive field next to that. So each column is coding for a different region of the visual space. Cells within a column are all coding for the same location in visual space. And the cells have overlapping receptive fields.
The columns are the different colored things. Like in other words, the white.
Okay.
Gray is one column, but why is there depth in both directions?
So imagine, that's a good question. I'm not doing a great job today. So imagine this is the surface, right? So this is a two-dimensional surface with a little bit of depth. The depth in this direction is the depth into the cortex. And as I was talking about it, you get multiple layers in this direction too. So is that clear at least that this direction is the depth of the surface? So this is the cortex surface and that's the direction. Is that clear? Okay. Now, what I'm showing here as black and white, these are ocular dominance columns. And these are showing that the signal is coming from the two eyes for this specific location. What does that mean? That means that, let's say this is a small part of the visual world. Let's just start from there. This is a small part of the visual world. The representation from the light from this part of the world hits which part of the left eye? If you remember from last week, it hits the temporal part of the left eye. It hits the nasal part of the right eye. Which one of them has to cross over to the other side? Which part? Temporal part of what? The what? So what happens if the temporal part of the left eye cross over? Where would it end up? Right hemisphere. But this is coming from the right visual field. Where should it end up? In the left. So which one of them would cross over? The nasal part of the right eye would cross over. Okay? So if this is in the right visual field, it means that the nasal part of the right eye crosses over. The temporal part of the left eye stays on the left side. And then what the brain does is that it actually gathers the information from the two eyes coming from the same location and puts them next to each other on the surface of the cortex. So the projection that we saw from the LGN to the primary visual cortex would actually keep information coming from the same area, but two different eyes next to each other. We talked about this. What is the benefit of that? What is the benefit of keeping the information from the two eyes next to each other in one hemisphere? And then what do you get from that? Depth perception too. So depth perception, how do you get depth perception from that?
If they're very different from one another, you know, oh, they're really similar.
Yeah, so if I'm looking at an object, if I'm looking at my finger and it's closer to me, the two images from the eye are going to be very different from each other. So just knowing how much discrepancy there is between the representation of the two eyes tells you how deep, how distant the object is from you. So that's one way. Now, okay. So basically the dark and the bright next to each other. Okay, so after that, going back to this. Location columns are a small patch, very small patch of the cortex that is representing the information coming from a specific location. And they're coming from the two eyes. So the projections come here, and that's the location column. If you move over to another location column in the cortex, you're representing a receptive field very next to that. So, and that's what we call the retinotopic mapping. Now, within each of these location columns, there are what we call ocular dominance column. Ocular dominance means like left eye or the right eye. How would you find this out? How would you find out what ocular dominance columns, if in the context of the experiment that I showed, how would you find out that, well, this area responds to the right eye or this area responds to the left eye? Yeah, so these are anesthetized cats. So you could just close one eye and then keep one of them open. The same light, right? The light is here, and you're not moving the location of the light. You're just closing one eye, opening another eye, and then recording from the brain and seeing where it is. And then when you close one eye and open the other one, you only have to move a tiny bit to detect the same signal, but coming from another eye. Does that make sense? They're right next to each other in the brain, in the visual cortex. But it's easy to study that. Now, within each of these ocular dominance columns, there's what we call the mini columns or the orientation columns. The orientation columns respond to a specific direction of the basically orientation of the light. Yeah, the lines are showing the orientation. All the way down the surface of the cortex. So the few millimeters that we talked about.
Same spot, same eye, same orientation for all 5 columns.
For yes, the rows here, yeah.
So it needs a lot of feedback. That one edge.
Feedback?
Well, it has 5 different cells, columns, or layers, sorry.
So layers, yes. So it means that the projection to this area carries information about the orientation, and all of them in this column are responding the same. So in my schematic here, if you look at these cells and each of them is a mini column, it means that all the cells, and from top to bottom, this is the surface of the cortex. This is white matter, which is the connections. And this is the surface of the cortex. So you can imagine, so if you take away this skull and everything, you'll see a surface. And then you put in an electrode and just going perpendicular to the surface in there until you hit the white matter, which is only, as I said, a few millimeters. All of those things that are in that path, the few hundred neurons that are in that path would have the same selectivity in the visual cortex. If they're responding to an orientation, they're responding to an orientation of light in a specific location from coming through a specific eye. So that's what the columnar organization of the primary visual cortex is. But knowing that, you could imagine that there are columns for every location in the space, for every orientation coming from every eye, for the two eyes. Does that make sense? So when I kept saying the word that it's tiled, That's what I mean, that these columns are tiling the visual cortex. And because the projection from the eye goes to the visual cortex, you get these basic feature detection across the entire visual field. So for example, if you have, let me go in this direction. If you have a curved line in a space, or let's say you have a straight line in a space, and the receptive field, and it's in the receptive field of this location column, and the receptive field of this location column, and this location column. These are three patches of cortex next to each other. How would this respond if there was a straight line in that space? Let's say a straight line, straight horizontal line. Which cells would be active? Okay, this, okay. What else would be active? What else? What? The whole line would be active. Because all of them, as I said, it is in the receptive field of all of these columns. Does that make sense? Why would that happen? Because, well, it is a straight line in a space. So, and if it is in the receptive field of this column, then it would only activate the features, it would only activate the column that cares about the straight lines. Does that make sense? This is what we talked about. It's a cell that cares about horizontal or vertical lines would not respond to that because it's not its preferred stimulus. Only this column would respond, but everything along the depth of the cortex would respond to it. The next one will respond to it because, well, it's there and it's coming from the two eyes. The next one, next one, next one. Again, this is not great showing, but imagine they were all similar. So you could imagine that the visual input, depending on its properties, would create a spatial temporal pattern of activation in the visual cortex. Does that make sense to everyone? So if it was a different stimulus, if it was a circle in a space, and this was a much bigger patch of the tissue, you would expect to create what? Pretty much a circle in the visual cortex. Because visual cortex has a retinotopic map of this space. And if it has a retinotopic map of this space, if you're looking at a circle, it would activate a circular area in the cortex. Does that make sense to everyone? Now, why is this feature detection? Well, in a way, because you have this tiling of this space, anything that's out there going to create a specific pattern of activation. It's not yet a circle. It's not yet perceived as a circle. It's just a pattern of activation in V1. It's only after multiple cascade of representation, then you perceive it as a circle. But initially, you have to have orientation information for everything, or orientation, and we talk about color and other stuff. You have to have these basic features from the visual space in order to build on top of it. Now, this is obviously a simplification when we're showing a square here, but this is what an actual surface of the cortex would be selected for. The orientation selectivity is color-coded. So horizontals are red and verticals are blue, and everything else is just the rainbow colors. And you know why that is. You know the wavelengths like changing from red, from infrared to ultraviolet, and everything in between is visible. What we see is that, well, this patch of the cortex, this is the surface of the cortex we're looking at. And this is pretty much this, yeah, this is just 1 millimeter. So you're seeing, well, there is dominance here. So this area is responding to horizontal ones next to it. It's not like a clear cut. So there is more of a gradedness that is changing across the board. But if you have a specific visual input, you would imagine that it would activate a specific part of this space that later on becomes interpreted as something. But initially, you need features. You need to, so when I'm looking out the world, different edges in the world are being processed by different patches of my visual cortex. Does that make sense to everyone? What does that mean? Like if, and I used another term here. I said different parts of the visual world are being processed by different patches in my visual cortex. What does processed mean in this context? Yeah, so they're translated to neural activity, exactly. So how are they translated to neural activity? Well, this part of the world has a lot of vertical edges. So there are neurons that like vertical edges in this part of the world, so they're going to be active. There are a bunch of horizontal ones in different areas. There are cells that like horizontal edges in that location, so they're active to that. So there is a complex, extremely complex pattern of activation happening in my V1 at 100 millisecond basis, right? As I'm moving in the world, as I'm moving my eyes or moving my head, right? The patterns on my retina changes. If the pattern on my retina changes, the pattern in V1 changes completely. It's only later on that patterns become stable and perception is built up. But earlier on, it's completely based on what the visual world is, and it changes with the new input from the visual world. Okay. So we call this area of V1 that is devoted to feature analysis of a specific region a hypercolumn. This is another term people use. It basically consists of one location column, two ocular dominance columns, and a complete set of orientation columns. That's a hypercolumn. And a hypercolumn could have maybe around 1,000 or a few thousand cells in it. Should we take a break? Or 7:06? But we've already had a break. And let's keep going, right? I don't think you seem all fresh. Okay, so the pathway that we talked about so far was a subcortical pathway. Subcortical because the cortex sits on top of the brain, and then everything below it is subcortical. It's subcortical because you have the input going from the eye to the LGN, which is again below the cortex, and then projecting into the primary visual cortex in the back. So we call these subcortical pathways. And they get input from the eye to the visual cortex. Now, when you reach the cortex, there are different pathways now in the cortex that we call cortical pathways. And again, pathway is not just a highway, but rather a cascade of neurons and connections that compute something. I talk about computation. Let me use this opportunity to give you an intuition about that. So far, we talked about, for example, well, I said this patch of the cortex processes visual input from certain part of the world. I could say it computes the edge information from certain part of the world. I could have said it that way, right? Oh, this patch of the cortex compute edges from this part of the world, right? So I could describe the behavior of the system in terms of, computation. And then another area computes something else. Another area computes something else. But you can imagine that processing is a form of computation. And it happens through chemical reaction. It happens through everything that we've learned so far in terms of, well, there was a spatial temporal pattern of activation in the eye because of the light that caused certain spatial temporal activation in LGN. That released neurotransmitters caused a certain pattern of spatial temporal activation in V1. And whatever activation is in V1 is basically the edge information in the world. So computation happens across these levels. Okay, So what are the main cortical pathways? So we're looking at the brain, and you could just, it should jump at you that this is the temporal lobe. The person is facing that way, and this is the back of the brain. So this is the occipital area. This is the parietal area, and this is frontal. So that's the bare minimum that you need to know, is that the back is occipital, up here is parietal, we have frontal, and we have temporal lobes. We have 4 lobes. The pathway that goes from the occipital lobe to temporal lobe is called ventral pathway. And the pathway that goes from occipital lobe to parietal is called dorsal pathway. And these are cortical pathways. It means that all the tissues involved in them is in the cortex. Now, people over the years have given names to these pathways and what they do, right? A lot of cognitive neuroscience and a lot of papers you'll read is about assigning jobs to tissues in the brain, right? That's just the name of the game. So you say, okay, this part does this, that part does that. So, and this is not different. This is basically people, and all these practices are inherently simplistic. All of them are simplifying beyond reason. So you should be very skeptical of them, but they're good to get an idea of what the brain does or what certain tissue does. But I'll talk about modularity a little later if we get to it today. But keep in mind that these are experiments and they teach us something, but it's never as extreme as they would want you to believe. So what people have thought the temporal pathway does is, the ventral pathway does, is that it's a what pathway? It's for complex pattern analysis leading to object recognition. And what the dorsal pathway does is for where? For analyzing what object locations are. And this was done, a lot of this in the early 80s. So why would people think that? Well, again, a lot of the early understanding that we have came from lesion studies. So if we lesion the temporal pathway and not the dorsal one, the ventral pathway and not the dorsal one, so you should already know that, well, this is going temporally, so it's ventral, and this is going to a parietal area, so it's dorsal. So if you knock out ventral pathway in a monkey or a subject or a human that you have, that comes with that damage, a lot of people have temporal pathway damage. Why would that be? What are the cases you think would someone come in with a ventral pathway damage? What could cause it? I was saying you could find people that have ventral pathway damage. It's not really an area that gets hit, like it's not like, sure, a football player, you get hit in the front. Like a lot of people get sports injuries in the front or in the back. But the temporal pathway, in the temporal lobe area, which is where the ventral pathway is, a lot of times seizures originate from there. So people have surgeries to remove part of their temporal lobe, which is basically part of the ventral pathway. But you could do this in monkeys, and you could ask them to do object discrimination tasks, right? Object recognition tasks. Well, how would you do that? You could put food underneath one of these objects, and then you could switch the area, but keep the association between the food and the object. So what the monkey would have to do is to recognize that the food is under this triangular striped shape, or it could be more complex. But what they have to learn is that this object is what is associated with food, not its location, because you could move it around across trial by trial. Then you could knock out the dorsal pathway, and then, well, it looks like the monkey is able to do this task, which is an object discrimination task. But if you knock out the ventral pathway, the monkey cannot do it. What does that tell you? What did these people infer from this study? Yeah.
What remembering where the object was in space.
Well, they're not supposed to remember it in this task, right? The reward that they're expecting is associated with object shape or object identity. So what they're learning, what they're supposed to learn is that this object identity is associated with reward because it's always underneath. For example, a peanut or a banana is underneath the triangular shape, even if the triangle was on this side. Does that the experiment make sense to everyone? The paradigm. So what they're learning from this experiment is that what judgment and object discrimination was disrupted by knocking out the ventral pathway, but not the dorsal lesion. Because again, the location can change from trial to trial, but the food is always under the triangular shape. So they have to look at it, whatever is in front of them, find the triangular shape, and then the reward is underneath that.
So it's like a double dissociation.
So this is a single dissociation. However, let's do another experiment and get to double dissociation. Well, what would we do in this case? In this case, instead of associating the reward with an identity, we could associate it with landmark location. Okay, so in this case, you're saying, well, the reward is underneath these things, but the identity doesn't matter. It matters that it's next to this other object. So they have to find the location of that object. It's a location-based thing. Now, you could see, well, people, when monkeys are the same monkeys with dorsal pathway lesion cannot do this task, but with the temporal lobe lesion or ventral pathway lesion could do the task. Okay, so what does that tell you? That tells you a double dissociation. That lesioning one area versus another area, or lesioning one pathway versus another pathway, gave you different outcome in terms of the behavior of the monkey. In one case, when the temporal lobe or the ventral pathway was lesioned, they were able to do the location discrimination, but not the object identity discrimination. When the dorsal pathway was lesioned, they were able to do the object identity discrimination, but not the location discrimination. Does that make sense? Yeah.
Yeah, do you mind saying exactly what you mean by object discrimination?
Rather than what?
So in this task, I have a tray in front of me, and there are two objects on it that are different from one another. And the reward, one of them is a triangular shape, one of them is rectangular shape. And the reward is always under the triangular object. But they could just switch them, and the reward next trial would be still under the triangular shape, right? In the location could change, but the reward is also associated with triangular shape, and the monkey has one pick. They could pick the triangular shape and get the reward underneath it. So they have to recognize, they have to recognize the triangle. Every time that, okay, I'm recognizing the shape that is triangle, and then associating that with the reward. Does that make sense?
Or it's like you're like talking about like the meaning that's often associated with a certain object, not just like distinguishing the object from other objects in space.
Well, those two things aren't independent.
Right, but I mean like
You have to distinguish
an
an object.
Not knowing that forgetting that the banana is underneath the triangle doesn't mean that you're not seeing the triangle as a triangle.
Yes, that's true. There is, so in an experiment like this, there is that the monkey would have to have like two things, right? They have to know the task that the object, that there is an association between an object and the reward. But in order to do that task, you have to recognize the object. You have to learn that, okay, these two objects are different, and this object is associated with the reward. But if you cannot dissociate the object identities, and that is what we call recognition, right? Dissociating object categories are what we call recognition. If I'm recognizing something as an apple, it means that I'm dissociating that category from other categories. And you have to be able to do that to do this task. You're right that you could do that and not learn the reward associations and not be able to do the task. That's true. But supposedly, if you take the same monkey and put it in this task, and the monkey can do it, you could show that the damage is not in the reward association. The damage is in not being able to recognize and dissociate two objects from one another. So great questions in a sense that what do I mean by recognition is something that it would have to take a couple of weeks for you to understand, I think, in terms of what do we mean. Like, it's a loaded term, but I think a recognition requires discrimination. So first of all, I think I mentioned this before. If I am able to do this task, so let's go through the task. It's important. The task is, well, I have two objects, and if I pick I have an apple and an orange in front of me, and I pick the apple, then I get a little reward. It could change the location of the object, and then I again have to pick the apple. It changed the location, I have to pick the apple. For me to be able to do that, first of all, I have the representation of the apple and the orange should be different in my head. Does that make sense to everyone? There has to be different. If they're represented the same, I'm not able to pick one of them. And at this point, think of recognition that way, that you have to be able to represent two objects differently, that their neural representation should be different in your brain. So you could say that the monkey with damage to their ventral pathway is not able to represent something in a way that allows them to do the tasks. That's the full answer. But for the sake of simplicity, let's say it cannot recognize the objects anymore. If it could recognize the triangle, it would associate it with the reward and they would be able to do the task. Okay? I think I'll get into the details of what recognition is in next week.
Yeah, maybe like a quick example of like the humans that have like damage to.
We'll talk about it. Well, in a few slides we'll talk about it. So people have revised this story of what and where. Now, hopefully you understand why this was called what and where so far, right? Yeah. Okay. People have revised this story of what and where to what and how, meaning that the ventral pathway is still largely associated with what is out there, like what is this object or what is that object, and the dorsal pathway is more associated with how do you interact with the objects in the world. I give you an example of a patient DF, who had damage to its temporal lobe, to her temporal lobe, which is in the ventral pathway, and she was unable to recognize object. So what does it mean to be unable to recognize object? In extreme cases, you show them an object, for example, an apple, as a model or a book, and ask them to draw it. In extreme cases, they're not able to do that. Like, this is an extreme case of visual form agnosia, where they're not even able to look at something and extract enough features from it to be able to reproduce it properly. If you ask them to draw from memory, they're able to do that. If you ask them to draw an apple from memory, they're able to do that, or draw a book from memory, they're able to do that. They're just not processing. When you put this thing in front of them, they're not processing it in a way that allows them to do the task. They're not capturing enough features in a way that, and this form of agnosia or this form of visual form agnosia is called apperceptive agnosia, which is very extreme, that people cannot really copy anything and they cannot name them. Like if you show them this object, they're not able to name them, right? If they were able to name them, they would draw it from memory maybe, but they're not able to name it. They're not recognizing it, right? And again, this idea of recognition, right? At some point you learned about apples, and now you're recognizing it as an apple. Like that's the idea. Now, this patient that has such a deep form of like visual form agnosia, they put them to this experiment. They asked them, just look at this. So they had an apparatus that had the apparatus looks something like this, which is a device that has a slot in it, and the slot could be a different orientation. So when they show them this and ask them, what is it? What is the orientation of this slot? Right? That's a very simple question. And the patient could not do that. So when you have such a damage to the temporal pathway, to the temporal lobe or the ventral pathway, you're not able to recognize, you're not able to identify what you're looking at well. You're not able to identify, well, this is the, orientation of this slot that I'm looking at. But interestingly, when the patient was asked to just take a piece of like cardboard or whatever you call this, and place it in this slot, they were able to do it. So the patient was able to actually interact with it in the right way without being able to recognize it, without being able to tell you what the orientation was. Now again, if you're a typical, functioning brain, and it's hard to imagine what this means, right? It's hard to imagine when people have lesions, how their brain works. But imagine that. Well, again, we know the data, so let's go with that. So when they were told to just identify what the orientation was, they weren't able to do that. But when they were card. When they were asked to put the card into the slot, then they were able to do that. They were able to interact with it. So this kind of was evidence that yes, the ventral pathway is for recognizing and identifying visual features, but the dorsal pathway that is not damaged is able to help this person interact with the object. So they don't have access to it. They don't have access to what the orientation is, but when they're supposed to act upon it, the information is relayed and they're able to act. Does that make sense? So the information about what the orientation is relayed through the eye to the dorsal pathway, and from there as results in the correct action, but they're not able to report on it.
Scenario just function like backup storage, if you will, like
Backup storage?
yeah, like simplifying it in the scenario, is it just functioning as a way to retain the information in the brain and complete the task, regardless of which one is damaged? It's like if the roles were reversed, for example, I assume that the wording for matching the card would work, but putting the card into the slot would.
Okay.
Is that true based on what you said so far, or is that incorrect?
No, I think you would still be able to do this task if the damage was to the dorsal pathway, but we'll have to find out. My impression is that because you're able to recognize it, you're able to correct for it even if you have a dorsal pathway damage. Unless the dorsal pathway damage is so strong that you have some sort of neglect and you're not even able to attend to it. So it really, these lesion studies are hard to interpret for those reasons because it depends on the extent of the damage and then it becomes whole other thing. But okay, this is a good opportunity. So brain has what we call degeneracy in a sense that you could arrive at the same solution from very different places. And in the brain, there are many pathways and they could compensate. One could compensate for another one. So when we have, for example, in this scenario, like you could, I would suspect if you have a dorsal pathway damage, you would still able to do this task through other means, right? The mean would be, well, I'm looking at the orientation out there, and then I could manage to orient the object in my hand the right way because I can recognize it. I can just push forward, something like that. So you could do this task. That's my expectation. But these are, again, these are good studies to get us thinking about these stuff, but they're overly simplified. What the entire brain area would do. Okay, so these people interpreted the data that they have as the ventral pathway devoted to feature analysis, and that have stood the test of time. Like up to this day, we think of the ventral pathway as doing feature analysis, as detecting all these visual information from the input. It starts from very low-level features like orientations and motion, and then it builds on top of that and becomes more complex features. The dorsal pathway has not been easy to analyze, and like this study over, I would say, up to this day, so people have found signatures of object recognition in the dorsal pathway too, or people have found signatures of a space or action or this or that. So the dorsal pathway has been a lot more debated and far less settled in its function compared to the ventral pathway. But also the ventral pathway has a lot more functions than what we're assigning it to here. Are you clear on the experiments that I discussed? I think they will be on the exam. Yeah, we discussed like maybe 3 experiments here. They're all clear, right? Okay, putting it. How is it that we're that behind? Okay, so putting this all together, we could draw a big picture of what the visual pathways are doing and what happens to the visual input as it enters the eye. So this is the view of the brain, and you have the front here, the back here, the brain stem, and you have the occipital cortex here, the parietal up here, and the temporal area here. We talked about that the light enters the retina, enters the eye, sorry, and projects on the retina. That's the first stage. The transduction happens at this stage. Then the neural activity travels to LGN. From there, it gets projected to V1, which is the primary visual cortex, and V2. This is the occipital cortex. Then it takes 2 different pathways. It temporally projects into the ventral pathway, and then dorsal pathway, it projects into the parietal cortex. And these are the 2 pathways that are shown here. The primary visual cortex, which is, let's say, V1 and V2, like the early visual areas, these are processing a lot of the basic information in terms of orientation that we talked about. in general, I mean, we'll talk about the other features that are segregated, but the primary visual cortex processes orientation. We talked about it. It's tiled in the space and it processes orientation on all sorts of stuff. Area V4 is in, so this is the ventral pathway as I'm showing down here. And area V4 has been shown to have a lot of, have an important role in color perception. So if you have damage to the V4 in one side of the brain, you may see the world in this way, that there's no color. So which side of the brain is damaged in this case? The left side, because the result is in the perception of the right side. And what you see is that you could see the part of the world that is being processed by the side of the brain that is damaged, the V4 area is damaged, as being without the color. So we call this hemiachromatopsia, which means hemi, you know, so far, it means like half of the world, kind of. Hemisphere, hemifield, we talk about it. And achromatopsia is the result of not having color in that part of the world. So people have observed that that patients with damage to V4 do end up with achromatopsia, which is the lack of color perception. So people have assigned this role to V4. It happens in the pathway, but again, go along with it like we're assigning that function to V4. Motion analyses, people have assigned that to PO and MT. Area MT is studied a lot for motion perception. So a lot of time motion perception is studied with these type of stimuli. You have a lot of dots moving in a space. Initially, they're all moving randomly, but a certain percentage of them are moving together in a certain direction. So imagine if it's 100% of them, it's very easy. If it's 50%, it's still very easy. But the monkeys could actually detect like 2%. If 2% of the dots are moving in the same direction and the rest of them are random, the monkeys are able to detect it. They're like, okay, this is the direction of movement. Now, if you damage area MT, if you lesion it temporarily with some solution or permanently zap it, you could see that the threshold increases a lot. Like it has to be 10 to 20% of the dots have to move in the same direction in order for the monkey to be able to detect the motion. So people have concluded that this area, and again, this is the core of all the lesion studies that we've seen, that if you zap out an area, if an area is damaged and the behavior becomes, you know, impacted, it could mean that this area was involved in that processing that behavior or generating that behavior. So that's the idea here, that the area MT is responsible for coding global motion perception. However, as you could see, the monkeys are still very good at this task. So when we talk about these sorts of modularity, saying, well, this area does that, that area does that, it doesn't mean that if you take that area out, suddenly this ability goes away, or that you're entirely, it really depends. There are other areas that are processing motion as well, and there are other cues that could be used. But damage to MT could cause impaired motion perception in humans. There is a condition called akinetopsia, and this is really the lack of motion perception. People with this condition perceive the world in terms of these, you know, sampled images across time. So for example, if you're standing by the road and a car is going by, you may not see the motion of the car, but you could see it every half a second that it's at a different location. So that's what we call akinetopsia. And this has been observed with people to damage the area MT. Motion blindness.
You showed the stimulus for the monkeys again.
Yeah, this?
So they're all moving, but 1 to 2% are moving in the same direction, or only 1 to 2% are moving?
No, they all should be moving,
But
otherwise.
they're kind of random.
Yeah. They all are moving. I could show you a sample of it next week. It's they're all moving, otherwise it would be too easy, right? They all should be moving in random direction. Then 2% of them are moving in the same direction. And from that, you could see, you could get a gist of it, you could get coherence. Sometimes you get that when you look at crowds that are moving kind of randomly, and a few of them are moving in the same direction, you kind of can detect that. In a playground, for example, the hectic playground, you could detect that. But yeah, that's the stimulus they use for a lot of the motion detection studies, because it's a nice stimulus because you could manipulate it easily, like how much of it are coherently moving, how much of it aren't. So it's good for studying motion detection. Okay, so we talked about V1, V2, and damage. We talked about damage to if an area of V1 is damaged, what would be the result of that? I think we talked about this last week, didn't we? Yeah, so you're gonna be blind, like there's certain area of the space gonna be very dark. Like you're just blind to it, like you're not seeing anything in that area.
So like, based on like this diagram and what I think I'm gleaning from like the other diagrams before, is this them suggesting that like later downstream, without sort of issues, like what's happening with like in V2 or V4 or whatever, like because V1 without even that basic information, that there would be challenges to like sort of all the other kind of functions in terms of color, sensation, or color like detection.
In what case, if there's a damage to V1?
Yeah, like, or like, because I'm just wondering, like, because right, I can imagine if you have damage in your retina, then I'm assuming that part of your cortex that's connected to that, like, you can't see anything.
Yeah.
So is that also true then? So if there's just damage, instead of the retina, but just damage to your V1 here.
Yeah.
The stuff under it is also impaired, or is it just?
Yeah, so as I said, if there is a damage in V1, then you just see a black patch there, like nothing, you're not going to perceive anything there. So people, and the retina is the same, right? A lot of degenerate, disease in the retina is that you start losing information in parts of the world. If you don't have photoreceptors, if the light is hitting your eye, the retina, and there's no photoreceptors there, it's just dark. Or other, diseases of the eye that could, for example, the liquid could thicken and then the light doesn't travel and then you see blurry, and so there are all sorts of things. But when it's that early, you see it, you perceive it as being a problem. You know what I mean? Like,
Yeah.
you see that it's blurry, you know that it's blurry, because it's so early in the vision. It's like looking through another person's glasses. You know that you're looking through a different lens. You're aware of it. So, we talked about damage to V1 does that. Damage to V4 has been associated with a lack of color perception. Damage to this part in the dorsal pathway has been associated with motion perception. Damage deeper into the parietal cortex, we've talked about this. This could cause what we call neglect. And if it's damage to one side, then it's a hemi-neglect, right? And we saw an example, we talked about it, where damage to the right parietal cortex could result in difficulty attending to the left visual field. And in this case, you're not aware of it. You don't know that your perception is missing something. And this is very different than earlier areas when you know that there is something missing. If you're not attending to it, and again, this is an extreme case, obviously, but in life, it happens a lot where you're not attending to something and you're not aware of it happening, right? So I was waiting for a package, let's say, and I go downstairs and I noticed that every 10 seconds there is a delivery truck going by. I've never noticed how many of them there are until I cared to wait for one. So I think we all experience this in life, that you suddenly attend to something and then you see, well, okay, I was now aware of it's happening. but take that to extreme and then you get neglect. Does that make sense to everyone? Like we've all experienced that attention is the first, like you've perceived it many times, but you haven't attended to that part of the world or that fact in the world or that predicament, and you've never, registered it. And this is really a matter of not registering anything in this part of the world. So we call that spatial hemi-neglect. Now, damage to this area, this is the end of the ventral pathway, an area called inferior temporal cortex. For short, we call it area IT, which is the initials of inferior temporal. The reason is that it's down here. So it's in the temporal lobe, but it's in the bottom of it. So it's inferior temporal. Now, when people look at the activity of IT cells and they want to study it with the same methods that we study earlier visual areas, so what do? Well, early visual areas, you were changing your light and you were seeing how does the patterns of activation change. Later on, it doesn't work that simple, right? You have to come up with much more complex stimuli to drive the cell, to actually get nice activation from the cell. And in this case, so far, I've shown you a lot of what's called raster plot, where you could see all the action potential. This is a different way of showing firing rate. Instead of showing a raster plot, you just bin it, and then you see how many action potentials are in each bin. So in this case, for example, let's say you have a lot of action potential in this bin, a little less here, a little less here. Here, you don't have any bin that has a lot of action potential. It's just a different visualization. And you could see that this cell, it's fairly active when there is a bigger object which is attached by a smaller oriented bar in this direction. So the idea is that, and we'll talk about this a lot more next week, is that as you go up in this hierarchy of areas in the ventral pathway, the selectivity of cells become much more complex. Because eventually you have to reach, for example, face recognition. There are cells that only respond to faces. What makes them only respond to faces, right? In V1, the cells are responding to different orientations of bars. What happens that across multiple layers of this hierarchy, you suddenly become only selective to faces? So there's an interesting hierarchy here happening, and that's the topic of next week. Okay, I just mentioned this. So this is stretching the topic, they're stretching this idea of columnar organization, but people have seen some degree of it in IT too. Again, because this is a cortical area, it's true of all cortical areas that there's just a thin, like, as I talked about it, area IT is also just a small patch in the surface. I should show you a figure of surface next week so you get a better idea of that. And you could also study that through the columnar organization and just go deeper in the column and see that, well, the cells that are within a column in this few millimeter within one column have similar shape representation. But again, this is a stretch in it. I don't think it's true to that extent. Again. So we talked about receptive fields. And I told you when there is higher convergence, the receptive fields are larger. So what is your expectation when you go higher in this hierarchy? When you reach area IT, what do you expect the receptive field to be? Much larger, right? Because there's convergence going on. Every layer, you have more and more convergence. So when you reach area IT, the receptive field is almost half of the world. So a cell could respond to visual input in half of the world. That's how large the receptive field is. What it gives it is location and scale invariance. What does that mean? Well, if you're changing with something, you're varying. So if you're changing the orientation of something or the size of it or the location of it, V1 cells will respond very differently to it. Because, well, if you change the orientation, they respond differently. If you make it bigger, the receptive fields are not hitting the same spots anymore. But if you have a really large receptive field, moving this, orienting it, making it bigger or smaller is not going to change the response of the cell because it's always is in the receptive fields. And that will be called location and scale invariance because the response of the cell is not changing based on those factors. It's invariant to the location and it's invariant to the scale of it. I don't want to rush through this, so let's just stop here and then we'll pick it up right here next week.
If you have not submitted the assignment for any reason, we're still missing a couple, please submit it and just double check Canvas to make sure it's submitted.